All right. Well, here we go. So this is section 19.7. I don't know why I had 16 on the brain, but this is chapter 19.7, cofactors and coenzymes. And this goes back literally to the first slide that we looked at this evening. I mentioned cofactors and coenzymes. So when we think about the active site of, or the active enzyme, sorry, an enzyme that is active, okay, that is ready to react, ready to do what it was designed to do. We call that a holoenzyme, okay? So that's just a name for, an active enzyme. Now, if you're wondering, like, why do we have to have a name for it if it's just, I thought the enzyme did the work and that's it. Well, it depends, okay? The part of the enzyme that is made exclusively from amino acids, so the polypeptide portion of the enzyme, that's called the apoenzyme, okay? So that's the apoenzyme. Whereas sometimes enzymes have a non-protein portion. If you're like, what? Yeah, they'll have a non-protein portion that's not made from amino acids. We call this a prosthetic group or a cofactor. So when we put the apoenzyme and the cofactor together, that makes a holoenzyme. Cofactors are bound to the enzyme for it to maintain the correct configuration of the active site. So what are we saying? We're saying you've got an active site in an enzyme, but in order for that active site to have the exact correct configuration, configuration is just a fancy chemistry word for saying in order for it to have the exact correct shape, it's got to have a cofactor bound to it. Examples of cofactors include things like organometallic compounds. These are compounds that are made up of just what they sound like, metals, combined with organic molecules. Metal ions, for example, like copper or iron or magnesium, and then organic compounds, which are oftentimes derived from vitamins. Okay, and we'll look at some examples of cofactors that come from or derived from vitamins. You can see why vitamins are important for your diet, right? So here's a representation of an apoenzyme and a cofactor. So it's not an apop enzyme. I just noticed there's a typo here. Let me scratch that out. Okay. So it's an apoenzyme, not an apop enzyme. Anyhow, so here's the representation of an apoenzyme and a cofactor. You've got your apoenzyme and you've got your substrate, like that. Okay. Here is the cofactor. Okay. In order for that enzyme to be able to do its function, okay, it's got to have that cofactor bound to it. Look at this very carefully. You see that the topography of the enzyme that I'm tracing out in red right now doesn't match the topography of our substrate, does it? Well, look, it's very clever how they did this. When you put the copper with the 2 pluses on it, all of a sudden, look, the topography here with the pluses and everything matches the topography of the substrate. So now you can see that with the cofactor bound to the enzyme, it's able to perform its reaction. A coenzyme, instead of a cofactor, a coenzyme is sometimes required by enzymes. A coenzyme is going to be an organic molecule. Okay, so a coenzyme, we'll just write in here, organic molecule. Now, what about that organic molecule? The coenzyme is going to be held to the enzyme by weak interactions, like, a dipole-dipole, or a hydrogen bond, or a hydrophobic interaction. Most coenzymes carry either electrons or small groups. So nothing big, just electrons or small groups. And many, again, have modified vitamins in their structures. Here's an example. Okay, now this figure is taken straight out of your textbook. Follow along with me. Look, in step 1, we have our substrate. Okay, let me get out the black pen. Okay, so we have our substrate right here, and then we have a second substrate over here. What you're going to see here is that the first substrate, you see that little red functional group on it? that red functional group is going to get transferred to the other substrate to form a product. And at the same time, when the first substrate loses the red dot, it's going to form a second product. Okay? Now, how the heck does that happen? The way that it occurs, it's not like the substrate binds to the enzyme and then this transfers the functional group. It doesn't work that way. You see how the functional group is round? Okay, well, look at this cofactor, this coenzyme. The coenzyme has a round pocket. And so what happens is both substrates bind to the enzyme, but the cofactor is necessary for the transfer of that functional group to occur from the first substrate to the second substrate. And then once that transfer occurs, they're both released to form the new product, which is the first substrate with the functional group on it. And then after the other substrate loses the functional group, it forms the other product, like that. So again, the coenzyme is kind of the key player here that actually works as like the mediator, right? It's passing the functional group from one substrate to the next. Here are some examples of coenzymes that are derived from vitamins. Take, for example, we think of an example that you would know if you've ever studied biology before, right? Earlier today, we talked about NAD+. That stands for nicotinamide adenine dinucleotide. That comes from vitamin B3. It's derived from that vitamin. Also, another coenzyme that you're going to see a lot later on in this course is FAD, flavin adenine dinucleotide. That's derived from riboflavin or vitamin B2. What else? What are some other good ones? You guys have all probably heard of coenzyme A. You've heard of acetyl coenzyme A. Okay, that coenzyme A is derived from pantothenic acid. I'm just looking here. Pyridoxal phosphate is mentioned in one of my videos. That comes from vitamin B6. Okay, it's involved in the degradation of amino acids. Anyhow, I digress. The point is this. You look at all of these different coenzymes, just the ones that I put in the red boxes or any of the other ones, all these coenzymes have very important functions in the human body. Where do these coenzymes come from? They're not just built out of thin air. They're built from vitamins. That tells you that it's very important that you don't have a vitamin deficiency in your body. This is why vitamins are so important, right? It's a big cascade, if you will. If you lack certain vitamins, well, you're going to be in trouble when it comes to doing, these functions over here. Anyhow, kind of interesting. Here's that NAD+. Remember NAD+? It was nicotinamide adenine dinucleotide, NAD+. And we're going to see this coenzyme come up several times throughout the course. But it says here that we're going to look at it in both its oxidized and reduced form. So follow along with me. Again, I'll never ask you to memorize the structure of NAD+. You can see it's got a pretty big, complicated structure here. But anyhow, NAD+ is a coenzyme that is of critical importance in oxidation reactions of the cellular energy harvesting processes like glycolysis and the Krebs cycle that we're going to look at in chapter 21. It's got this big structure. Again, I don't expect you to memorize that, but look at this one point of the molecule that they want you to focus in on. Now, you guys all know bond line structures, and you know that there's a hydrogen attached to this carbon. But NAD+ can be reduced by a hydride. Remember, H+, that's a proton, right? H-, okay, that's called a hydride. Okay, so when NAD+ takes on a hydride, and here's that hydride, it becomes NADH. So NADH is the reduced form. How do we know this is reduction? It goes all the way back to, one of the first chapters we looked at. What did we say? We said that reduction in organic chemistry is gaining hydrogens. So since the molecule is gaining a hydrogen, it's being reduced. And if you're like, well, is there a reverse process oxidation? Absolutely, right? Because you're losing hydrogen in the reverse process. Again, you can see how the coenzyme is a pretty complicated molecule, right? So a lot of elements in this molecule. This is just another representation of NAD+. It says the nicotinamide part of NAD+ accepts the hydride, okay, plus 2 electrons from the alcohol to be oxidized. Now, I'm not that concerned with electron counting here, but I just want to show you that what you learned in Chemistry 101 is still true, okay? You probably learned oil ring. Oxidation is losing electrons and reduction is gaining electrons. Look what the hell is happening here. Not only is the NAD+ gaining H+, but it's also gaining 2 electrons in the process to form NADH. So if it's gaining electrons, right, that's reduction is gaining electrons. So this is the reduction process. Okay, so that's why it's being reduced. All right. These are 2 other adenine dinucleotide coenzymes. One is NADP plus, or NADP, yeah, NADP, or FAD. And you're going to see that these are both used in redox reactions that we're going to look at in chapter 21. Again, I don't expect you to memorize their structures, but you can see that the NADP is closely related to NAD, except we have a phosphoryl group on it here. And FAD is flavonoadenine dinucleotide. This is the oxidized form here. And if you're wondering, like, what the heck are these pink arrows about? Well, the pink arrows are where there's obviously a hydrogen here. There's no hydrogen here, and there's no hydrogen here. But this is where the reduction occurs, okay? So when their FAD is reduced to FADH2, it adds a hydrogen here and another hydrogen here. And when NADP plus, or NADP, is reduced to NADPH, the hydrogen is gained here. Again, these are things that people who have taken biology before, I'm sure that you've all heard of the reduced form, which is NADPH, and the reduced form of FAD, which is FADH2. It's probably nothing new to those people. And if you're like, hey, it's new to me, well, again, we're going to look at these even more in chapter 21. But the take-home message from chapter 19 is that these are coenzymes. And if we back up a little bit, okay, where did it say it here? Something here, okay? That most coenzymes carry electrons or small groups, okay, like a hydride, okay? Nothing, you know, gigantic. All right, let's get back to where we were and talk about environmental effects on enzymes. Remember, we spent some time talking about denaturation of proteins in chapter 18. And since an enzyme is a protein, you can guess that a pH will affect the reactivity of an enzyme, won't it? The environment surrounding an enzyme can have a direct effect on the enzyme function. Enzymes, in fact, work best within a particular pH range. Extreme pH changes will denature the enzyme. That just destroys it. Then it's not going to be able to do anything. Some examples of enzymes that have optimum activity in different pHs would be pepsin and trypsin. They're both proteases. They're both enzymes that catalyze the hydrolysis of proteins. But pepsin works in the stomach, and it works best at a low optimum pH. Why? Because the stomach is very, very acidic. It's got a pH of around 1 or 2. Whereas trypsin, which is found in the small intestine, has a high optimum pH because the intestines are basic. And so what you might see with some enzymes is that, you know, here's the rate of reaction. It'll increase, you know, at a lower pH, and then all of a sudden it's going to attain a maximum value at a certain pH. Well, what's physiological pH? Physiological pH is around 7.4, right? 7.35 to 7.45. And then what you're going to see is that if you keep increasing the pH, is that the activity of the enzyme is going to dramatically drop off. Here's a picture that I took from another textbook that shows pepsin and trypsin. You can see they're very big, you know, complicated molecules, these enzymes. And the optimum pH for the activity of pepsin is around 2. That's strongly acidic, right? This is very acidic, but the stomach is acidic, isn't it? whereas trypsin's optimum pH is around 8, which is basic. Okay, but again, as I told you earlier on, the pH of the intestine is in fact basic. So different optimum pH levels for the rate of enzymatic reaction or enzyme action for different enzymes. Does temperature play a role in enzymatic activity? And the answer is absolutely yes. An enzyme has an optimum temperature associated with its maximal function. If you think about uncatalyzed reactions, okay, so these are reactions that don't have anything to do with enzymes, right? What I told you is kind of the rule of thumb is that every time that you increase a reaction by 10 degrees, you go from 10 to 20, normally you get a twofold increase in the reaction rate, so you double the rate of reaction. Well, what about an enzyme catalyzed reaction? The optimum temperature is usually close to the temperature at which the enzyme typically exists. So that's going to be body temperature, or 37 degrees Celsius for humans. Well, what's going to happen if we crank up the temperature? That's going to cause the enzyme to denature, right? That's something that we discussed a lot earlier on in this class, right? Say this is your optimum temperature right here. You start cranking that up, you're going to get denaturation, which is going to render the enzyme completely non-functional, destroys its function. Okay, let's switch gears and talk about how do we regulate enzyme activity, right? Why are enzymes not just like the Wild West and just react with anything at any time, any place, anyhow, anyhow, right? Well, we discussed that a little bit already in terms of enzyme specificity. But once we know that an enzyme can react with a specific substrate or a specific type of bond or whatever, how is the enzyme activity regulated in the body? Well, one of the major ways that enzymes differ from non-biological catalysts like hydrogen, like palladium, platinum, or nickel in our hydrogenation, for example, is the regulation of biological catalysts by cells. Some of the methods that organisms use to regulate enzyme activity are, first of all, to produce the enzyme only when you need it. Right? This is common in bacteria. Why would a living organism spend energy, which is precious, to make an enzyme when it's not required? So that's one way to regulate enzyme activity. The next one is for the enzyme to be something what's called an allosteric enzyme. We're going to look at that right now. Another way to regulate enzyme activity is to use something called feedback inhibition or a zymogen, and then finally protein modification. Well, we've already covered the first one, to produce the enzyme only when the substrate is present. Let's take a look at allosteric enzymes and discuss what that actually means. Okay, here we go. So an allosteric enzyme, it says here that an effector molecule can change the activity of an enzyme by bonding a second site. So an allosteric enzyme is really nothing more than an enzyme that has more than one binding site in the molecule. It's got two binding sites, right? A second site. Now there's two types of allosterism. We can have positive allosterism and we can have negative allosterism. Here's a cartoon or a figure that represents positive allosterism, which is where the effector binding converts the active site to an active configuration. Here we go. So you can see that we have a substrate that wants to bind at the active site, but the active site is unavailable. So what positive allosterism is that there's an allosteric site in the molecule. And a second molecule called a positive allosteric regulator is going to fit into that allosteric site. Okay? And when it does, it's going to change the conformation of the enzyme such that it's going to open up the active site for the substrate to enter. So a positive regulator changes the activity site or the active site so that the enzyme becomes a better catalyst and the reaction rate is going to increase. Negative allosterism, which is shown down here, is where an effector binding converts the active site to an inactive configuration. Here we go. Look, we have a beautiful active site. You can see the topography matches the notch in the substrate. Everybody's happy. Well, what happens is The negative regulator is going to bind to the allosteric site and it's going to shut the door, right? All of a sudden it's like, I can't bind anymore. And so the conclusion is that the negative regulator changes the activity site so that the enzyme becomes less effective and the rate of the catalysis slows down. Now, our book, for some reason, she doesn't have any figure that shows positive allosteric control or positive allosterism. But she does have a figure that shows negative allosterism, and it's this figure right here. This one comes from our book. It's even more elaborate than the example that I just showed you. Okay, effector molecules change the activity of an enzyme by binding at a second site. And then here, the example of negative allosterism or feedback inhibition. Let me show you what's going on here. This is a big old enzyme, isn't it? This enzyme is so big that it's got, different subunits, 1, 2, 3, 4. And then we have a binding pocket right here for our substrate. You see that the substrate is bound in there. And then what's happened is that the enzyme the substrate is being converted not into one product, but into two products. Look at these. You got first product here, but this second product here has an interesting shape. And it just so happens that that second product is going to be able to fit in the allosteric site. But since we're dealing with negative allosterism, what is it going to do? It's going to shut the door on the active site, right? You can even see the lines here, right? It closes the door. And so now the binding of the effector in the effector binding site causes a conformational shift, right, of the enzyme that closes the active site and inactivates the enzyme. Now, if you're wondering, like, well, now that it's shut down, is that the end? Is the enzyme never going to work again? Well, think about it this way. If this product dissociates from the enzyme, it's going to reopen the enzyme so that it's going to be able to produce more product. Okay, here's an example of allosteric enzymes and metabolism. This is a reaction that we're going to look at in chapter 21. It's the third reaction in glycolysis. Again, for the people who have studied biology, I'm sure that you looked at the steps of glycolysis and the Krebs cycle, and you probably would have heard of some of these enzymes. But now that you've studied organic chemistry, it gives you an appreciation for, what are the structures of these molecules? What the heck is actually going on here? So the third reaction of glycolysis places a second phosphate on fructose 6-phosphate. Sometimes we abbreviate this F6P. Here we have one phosphate, and ATP has a phosphate group that gets transferred onto this hydroxyl here. So now we have two phosphates on here. So instead of being adenosine triphosphate, it becomes adenosine diphosphate. The enzyme that does all this, wonderful business here is called phosphofructokinase. Why do we know it's a kinase? Because it transfers a phosphoryl group, right? We learned that at the very beginning of the lecture this evening. But what I want to think about with you guys are these two little molecules, ATP, adenosine triphosphate, and phosphate. If we think about the progress of this reaction going from F6P to fructose 1,6-bisphosphate, these two molecules, ATP and ADP, ATP is a negative effector. Now, ATP is kind of the currency of energy in the human body. If we have an abundance of ATP in the body, why would we need to metabolize or break down a sugar, right? Why would we need this reaction to occur? The answer is we wouldn't need that reaction to occur. And so ATP is going to slow down this process. Whereas AMP, which is a derivative of ADP, if I need to metabolize more sugar, what AMP is going to do is it's going to act as a positive effector, and it's going to cause this reaction to speed up. Okay, so positive allosterism by AMP, which again is a derivative of ADP.